I want to start by thanking the organizers and the Australian Immunization Coalition for inviting me to talk at this symposium. I am sorry that I'm not there in person. I'm going to be talking about recent pandemics and informed predictions. I think that there are several in the room who have thought about this at least as much as I have, if not more. And unfortunately, I'm not there to hear their opinions. So but the kinds of pandemics I'm going to talk about are mostly respiratory. I think if we look over the last many years, the pandemics that come to mind are in human populations. There's the 1918 flu pandemic, the H1N1 influenza pandemic in 2009, the 2019 COVID-19 pandemic, and various Ebola outbreaks and epidemics, and the HIV pandemic. And of note, the ones that were most intractable to deal with were respiratory virus infections. And though, of course, HIV has proven very, very difficult to deal with. And all these infections are zoonotic, meaning they came from animals. So the first prediction that I'd like to make, and many of these predictions are more commentaries on where we are as much as predictions. So the first prediction is that the next pandemic virus will be a zoonotic respiratory virus, but with one caveat that I'll mention. Humans will be immunologically naive to the virus, and virus needs to be able to officially transmit from human to human. This was really a key thing when we consider the SARS coronavirus and SARS coronavirus 2, since even though both use ACE2 as a receptor, but only SARS-CoV-2 could efficiently transmit from person to person. And why this occurred, we still don't know. And I think this is really a key thing we need to find out, because these two viruses seem so similar on the surface. This will be a novel zoonotic virus, but there's also some caveats we need to think about, and this comes from the field of feline coronaviruses. So there's two feline coronaviruses that are worth mentioning. So one is really a canine virus with a feline component. This was identified in children in Malaysia with respiratory illness. It contains genetic information from canine, feline, and swine coronaviruses, and these viruses all use the same receptor, so the viruses can easily cross species. This was never found in nature. There's no evidence for interhuman transmission, but we don't know if that's going to change. Now, the second coronavirus is actually more worrisome, because this is a virus that was found in cats in Cyprus. So the feline coronavirus normally is an enteric pathogen. It causes diarrhea. It's usually cleared, but some cats become persistently infected. In these cats, the virus changes so that it now, instead of just infecting cells in the lining of our mucosa, the epithelial cells, it also infects macrophages. And the name of this virus now becomes feline infectious peritonitis virus because it causes a lethal disease, FIP. Oops. And the thing about this virus is, because it only arose in persistently infected cats, it doesn't transmit from cat to cat. And cats are not immunologically naive in this circumstance. Now, what occurred on Cyprus was a new FIPV, and the thing that's scary about this was a recombinant between feline coronavirus, that's shown in the red box, and this little bit of another canine coronavirus spike protein. And this virus can now transmit from cat to cat. So because of that, it's highly virulent and transmissible. So there were 8,000 cats that died in Cyprus. This virus has been found in the UK, and it certainly has the potential for being a pandemic virus in cats. So the second prediction is that surveillance will be critical for identifying potential pandemic viruses. We've learned a lot about this over the last few years, and surveillance has to be done with a One Health approach. So what does this mean? It means we need to do surveillance at the human-wildlife interface. We need to do surveillance for human and wildlife antibody responses to high-risk pathogens, even if we can't find those pathogens. And we would do that in areas of high zoonotic crossover. And we need to survey patients with unexpected or undiagnosed pneumonias in high-risk areas. Not undiagnosed, but pneumonia for which we don't have a specific etiological cause. And the surveillance has to be focused, otherwise we're going to get information overload. What do we do with this information? We can think back to 10 years ago when there were studies that showed bat coronaviruses could infect human cells, and these turned out to be similar to SARS-2, but what could we have done with this information? Would we have developed vaccines, antiviral therapies? It seems unlikely that either approach would have received monetary support prior to 2019. Even the MERS coronavirus vaccine for a disease that we know infects humans with a high mortality really didn't receive adequate funding prior to the pandemic. And I don't know how the funding is now for MERS-CoV, but I bet it's not where we need it to be. So in the beginning, I think the other thing that another prediction is we're going to know more about the agent that causes the pandemic than we think. In the beginning of this pandemic, of the COVID-19 pandemic, there was a fear of the unknown, especially with the 2 to 3% mortality. However, we knew a lot about the replication strategy of the virus, and reverse genetic systems were well established. And we knew some of the targets for antiviral therapy. There had been many groups working on protease inhibitors, and these had been developed for FIPV, the disease I just mentioned. And these were very good. They reduced mortality in cats from 100% to 50%. And there was also many vaccines that had been developed for coronaviruses that cause disease in farm and companion animals. Many of these didn't work well, but they guided SARS-CoV-2 vaccine development. In specific, we knew that the spike protein was really the target for the neutralizing antibody response. So I already mentioned this, and we also knew something about T-cell responses from these studies. However, with a new pathogen, it'll be important to quickly identify targets for the immune response. We may not be so lucky, and the spike protein may not be sufficient as a target to neutralize the virus. Another thing we knew how to do is monitor virus evolution. This was based on influenza virus studies, and we knew that coronaviruses caused immunopathological disease. And I'll show that an example of this from my own laboratory. This is an infection of a mouse with mouse hepatitis virus, and one could see as one infects a mouse that has no T or B cells, that one gets virus all along the spinal cord in this case, but the myelin over the spinal cord is perfectly normal looking, and the axons underneath look perfectly fine. If one now adds back T cells, very quickly the virus is cleared, but at the same time, the myelin is destroyed. So this was always a key thing about coronaviruses. How come they couldn't be cleared without causing disease? And this is what immunopathological disease is. The immune response is causing the disease and damage. So another prediction is that we will make mistakes, and some of these were medical. So early in the pandemic, it took a while to realize that the pathophysiology of SARS-CoV-2 was a little different than other virus infections. We use ventilators a lot, and these created their own problems. And it took a while to realize that with this disease, use of ventilators should be delayed or not used at all. The patients needed to be prone in order to have best outcomes. And we had an initial understanding of transmission was incorrect. We thought it was going to be large droplet, or contact, but in fact, it turned out to be aerosolized, which meant that masks would be useful, something that we didn't realize right away. And this comes to the next thing, is we will make mistakes in communication, no matter how hard we try not to. Communication with the broad public was often difficult. Errors were made. Many of these occurred because new information was available almost on a daily basis. For example, one example that affected me directly was I often was asked to comment on aspects of the pandemic, and early on we thought that pregnant women had no more severe disease than non-pregnant people, but then it became clear that in fact, even though fetuses were not infected, the placenta could be. In pregnancy outcomes were a little worse. And finally, it was realized that pregnancy was a risk factor for worse outcomes. So that pregnant women were ending up in the ICU and dying. And this is one of the reasons that we strongly recommended vaccination when it was available for pregnant women. Another example was mask usage, which I think we all know about, and I just briefly mentioned how it was recommended and has not mentioned to be not important initially, but as the pandemic progressed, to realize it was important. School closures were another issue that I think has been discussed. And what we can say from all this is that communication with the public needs to be nuanced and be correct, so not overstate what we know. Then another point is we'll make effective uses of scientific advances. Most prominent one in the present pandemic is the use of that for vaccine development, where we used advances in RNA technology, vaccine formulation, adjuvants, and to make better vaccines. And as part of basic research, several groups led by Barney Graham and others developed pre-fusion surface glycoprotein constructs. This matters because the virus coming into a host will actually be pre-fusion, might look something like this, and after binding to a cell, it'll look like this. These so they clearly have different patterns. The tendency of the virus to actually undergo more structural change to become post-fusion, even if you have it in the test tube, hindered the development of vaccines. So this kind of understanding that we want to keep the protein in this form really made a huge difference in developing the vaccines. And this was all because we had scientific advances prior to the pandemic. And so what kind of advances might be useful in the future? What we learned is that we actually don't know that much about the correlates of protection, and they seem to be changing as the pandemic is changing. But we know we've learned it's important, and we were lucky in that this pathogen correlates really focused on the neutralizing antibody titers, though they are definitely not the whole story, because we know now that a lot of the variants that we're seeing now are not recognized by the immune response induced by vaccines, but people are still highly protected. We've learned a lot about the maturation of the antibody response and memory B cells versus plasma cells. And we've also learned about T cells being important in protection and non-neutralizing antibody function, as well as memory innate cell functions. So I've gone over this very quickly. I'd be happy to talk with anyone who wants more information later. But these are some of the things that we've learned. We've also learned to use wastewater to monitor virus spread. This is especially important when testing in patients is diminished. And we've started to use human challenged studies. And so what are the other things? Another prediction is that future pandemic will bring out the opportunists. This seems pretty clear since it certainly occurred in this pandemic, the COVID-19 pandemic. So just recently, this is from just this month, the Florida State Surgeon General called for a halt in the use of mRNA vaccines because of the risk that DNA integrated into sperm egg gametes could be passed on to offspring of these vaccine recipients. This, I don't know. It's hard to know whether this person is saying these things because he power issues or because he really believes this. Either way, he's the head health officer in Florida, so people listen to him. So this is the kind of things that we've been dealing with. The other way that people use or act as opportunists in actually in financial reward. So many anti-vaccination websites say don't get vaccinated, but what you should do is use unproven therapies such as ivermectin or something or anything else, which they will happily sell you. So this is a second thing. So we learned that it's important for all of us, public health authorities, scientists, etc., need to actively refute these claims. Next prediction is that there'll be a wider availability of vaccines and therapies worldwide, and this is hopefully will occur because countries like India are now able to produce vaccines and therapies at less cost than is seen in the U.S. and other places, and hopefully this will ameliorate some of the global inequalities in terms of vaccine, and then there's organizations like CEPI, which are stockpiling viruses. The issue of vaccines, the problem here is that both vaccines and antiviral therapies expire. So we never really know what to stockpile and when they'll actually be needed and what the next pathogen will be. But the one thing, as I mentioned already, though, we learned that you can make vaccines rapidly and safely. So maybe stockpiling isn't the key thing to do. Just being ready to go is the key thing. And one thing that we certainly have learned in the US and I think elsewhere as well, it'll be critical to overcome vaccine hesitancy. And then, so this is aspirational. There'll be cheap and readily available antiviral therapies. This would be a good way for things to play out if there was a cheap oral antiviral therapy. But this is really hard because, as I mentioned already, it could well expire, so it wouldn't necessarily be useful for long periods of time. A single protease inhibitor might be active against many coronaviruses, so it'd serve to be almost like the kind of universal coronavirus vaccine that we've talked about. And it would need to be nearly free to work in low income countries. So I'm just going to do one thing here. Okay. And so the second aspirational statement is that there'll be differences in access to vaccines and therapies depending on ethnic, racial, and economic factors, and differences in acceptability, but that we will be able to overcome these. And this, I think we're getting better in the U.S. Many of these disparities in vaccine uptake seem to decrease. Initially, populations that were more skeptical of the health system, such as mostly African Americans and Native American Indians, they were somewhat skeptical, but they have had much more uptake than with time than they had in the beginning, as this is opposed to white American populations, which exhibited decreased vaccine uptake. So they became more similar in their efficiency of uptake, though not necessarily for the right reason, at least on the white American side. Uptake in age populations was not a critical factor in the U.S., but is in countries in Asia, such as China. We know that that's what contributed to the large number of deaths that occurred when China opened up. Aged individuals had not gotten vaccinated. And even now, I have Chinese postdocs in my lab whose parents and grandparents, or mostly grandparents, refused to get vaccinated. So the next to last prediction is that there'll be human altruism. And this was really clear during the pandemic, where we had people really working huge numbers of hours. I think this picture is from Australia, and maybe one of these others as well. And it's not only physicians and nurses who worked extraordinarily long hours, but these are people, some of the ancillary help, who did various things, who were very critical for dealing with the pandemic. And then there were people who were outside of healthcare who also were on the front line, people who were bus drivers or custodians, police and firefighters. They were always at risk for many of these for the early infection. And so they have to be considered altruistic for continuing to carry on. In research laboratory, many places actually shut down for the pandemic, but animal facility personnel and some lab workers came in, again, partly because they wanted to do their work, but also partly because they knew that they had to carry on and keep the laboratory research going on. And then finally, this may be the only real prediction that's novel or new, maybe it's not new, is that if the next pandemic is caused by a coronavirus, it won't be a Sarbeco virus, which is the kind of virus that SARS-CoV and SARS-CoV-2 are, because I'm much more worried about MERS coronavirus, which is the cause of Middle East respiratory syndrome. This is a virus that's a camel virus. It rarely spreads to people, and it rarely spreads from human to human, except in the hospital, maybe household. It has a mortality somewhere between 20 and 35%, so a bad infection, but luckily it's so non-contagious that it just doesn't really cause human-to-human disease. But could it become more transmissible? We saw already with the feline infectious peritonitis virus that these things happen without our expecting it. And the other thing is that we learned in COVID-19, we thought a lot about SARS-CoV. We started developing vaccines, but that turned out not to be the problem. Rather, it was a virus related to SARS-CoV. So there may be SARS-like coronaviruses, and some are starting to be identified in bats and camelids and perhaps other animals that should be high on the list of animals that require surveillance and vigilance. And so the other thing is that even MERS coronavirus has some tricks up its sleeve, because MERS-like coronavirus can also use ACE2, which is the receptor for SARS-CoV-2 and SARS-CoV. So there's a lot of pathogens out there. We have to be really vigilant, and but we have to be ready for more than just a Sarbeco virus. So these are the people in my lab, and I only show this because we got about 2/3 of a meter of snow last week, and The people in my lab may be crazy going out in short sleeves and shorts, but hey, that's the way it is in Iowa. Okay, thank you.
How does the scientific community have open and transparent discussion about its developing knowledge without the community losing confidence and the apparent disagreements that we have about known unknowns?
Well, I think that's a hard question to answer. I think that, to my mind, this is all we have to have those discussions and make clear what the discussion points are, and even to the wider community. I know this is a trick because especially the larger public may want answers that are more absolute than, as scientists and healthcare personnel, we feel comfortable giving. But we just have to figure out how to do that. Because if what we learned from the pandemic is we can't be adamant because we're often not right. So we have to be nuanced. We have to make clear that what we know and what we don't know and where the discussion points are. That's the best I can answer.